Okay, so we are going to talk about analyzing bulk RNA-seq experiments today. We're going to go over the biology of the steps in bulk RNA-seq. We're going to interactively submit a job to use our pipeline called CHIRP to analyze bulk RNA-seq data. We're going to get a good understanding of what the outputs should be as well as the inputs and then talk briefly about some of the next steps. So hopefully by the end of this tutorial, you'll have a good grasp of what the necessary inputs and outputs are, know how to use our in-house pipeline. And I would just like to mention that this pipeline has been published many dozens of times. It's what we use as analysts to analyze bulk RNA-seq data. And also, if you have a simple bulk RNA-seq experiment with just a treatment and control sort of a design, we will even analyze that for you if you start a help desk ticket by sending us an email at help@msi.umn.edu. We're going to talk about some important quality metrics and show how to identify differentially expressed genes. What we're not going to be doing is we're not going to be giving a detailed Unix or Bash tutorial on all of the possible commands and ways that you can navigate the command line. I will give a very brief bit of information about that in order to know enough to submit the jobs today. Similarly, while we are going to submit jobs really, on MSI, on the supercomputing cluster, this is not a detailed guide to job submission. We have other tutorials for that. And then also, we are going to be using a pipeline which we've sort of painstakingly put together from many different pieces of software, each of which have their own parameters, which could be modified and which you can actually modify with CHIRP. And we're going to talk a little bit about that, but we're not going to go into extreme detail about all of those sub-programs parameters. And then finally, this is about bulk RNA-seq. So when you take some tissue, you grind it up, and you've got many different cells RNA all mixed together. This is not about single-cell RNA-seq. We have a different tutorial for that. It's also not about small RNA or differential splicing. But if you're interested in pathway enrichment, for example, we have that tutorial 1 week from today, and it follows directly from this tutorial. And if you are interested in single-cell RNA-seq, we have that tutorial 2 weeks from today. And that one's been revamped pretty significantly, so I'm excited to see what Natalia does with that. All right, so all of you should hopefully have the main prerequisite for this tutorial, which is access to MSI's Agate cluster. When you signed up for the tutorial, you were likely given a login for MSI's systems, or you already had one due to the lab that you run or are in. We'll go over some command line navigation, like I said, just enough to get us there. But if you want more information, there's this great Intro to Linux page here made by one of our colleagues. For those reading the tutorial later on, there's some formatting information that I'm not going to bother going over right now. All right. So this graphic we put together to show at a very broad bird's eye view what a bulk RNA-seq analysis looks like. It, of course, starts with some sort of biological experiment and a question. So in this one drawn right here, the question being asked is, how does gene expression in these mice change with treatment? And in this small example, we have a grand sample size of 4 mice, which is a little n of 2 per treatment. We've got 2 treatment mice and 2 control mice. So it's replicated. It's the minimal amount of replication. We always recommend 3 or more, but I just show 2 here for this graphics. It makes it easier. And you start with that. You run some sort of experiment where you're expecting or at least hypothesizing for some effect on gene expression to occur. Then in the lab, or sent out to a company, you do some sample preparation. You extract the tissue that you're interested in, and from that, you extract the RNA. You convert that using chemistry into cDNA, complementary DNA, and you usually add some adapters, and you end up with a tube for each organism that has many, many millions of different RNA fragments which have been converted into this cDNA and with adapter sequences added to them. So just like we had 4 samples there, we have 4 samples at the end of sample preparation. At that point, you send those that cDNA off to a sequencing company such as our core here, the UMGC, and that your fragments will get read on some sequencing machine. That could be like what we've got here, a VD, could be an Illumina-based machine, could be something else. Here we're going to focus mostly on short reads and paired reads, but there's multiple different ways that this could occur. Once your data gets sequenced, if you did paired end sequencing, which is what we're going to talk about here, you will have 2 files as a result per each sample. Those are called FASTQ files, and they will contain for each fragment that got sequenced by the sequencing machine, its best guess as to what the sequence was in that fragment read from the first direction, and in the other file read from the other direction, as well as information on the name of the reads, and finally, importantly, quality information as to how well it thought it could make a call for each base in its sequence. And we'll show one of these in a little bit. And so that all of that part there, the sample preparation and the sequencing, is outside of the scope of this tutorial. UMGC is a great resource for help in understanding how to do the sequencing preparation and sequencing itself. If you do have questions about experimental design, however, you are welcome to email us here at MSI, because oftentimes we'll analyze experiments, and so therefore we really like to help people come up with a good, solid experimental design that can help avoid some pitfalls like confounding. So then, after all that happens, you end up with these FASTQ files per each sample, and now you need to do something with them. Well, probably what you want to end up with at the end is an analysis where you have, for example, a list of genes which may or may not be differentially expressed between your treatment and control, perhaps some unsupervised heat maps which show which genes are higher and which genes are lower, and also what the variability is between your samples. And to get there, you've got to get first to this very important thing called a counts matrix. And so what this matrix is a rectangular matrix where each row represents a different gene, and then each column represents the number of fragments from that sample that mapped to that gene. And so we can see in this particular example, this first gene had no counts in any of our samples. Then this one seems to be lowly expressed. This one's more highly expressed, etc. We also kind of see, importantly, that the sums of the counts per each sample differ, and that ends up being important downstream when things get normalized. We won't go into total detail about that, but it is important for the statistics. And so we need to get to this counts matrix from our FASTQ files. And that's largely what the bulk of our CHIRP pipeline does. It does the sequence analysis to get from the raw FASTQ files to the counts matrix. Then the second half of the CHIRP pipeline goes from that counts matrix to our actual analysis. Okay. I'm going to go through this on the together, so I'm going to skip that little bit, but I want to point out very importantly right here, this link to this text file right here has, hide this window, okay. This window just keeps wanting to pop up. There we go. This link right here goes to this text file right here, which contains all of the commands that we are going to run. And so this is a really nice cheat sheet for running your own RNA Seq pipeline using CHIRP. There's going to be a couple of very minor changes due to dates and the fact that you are not me, but otherwise this is going to be more or less exactly what we are going to run. All right, so we know that sequencing ends with FASTQ files, and that's where this tutorial begins. So let's go and look at some sequencing files. To do that, we're going to use MSI's amazing tool, which was originally created by the Ohio University, called Open on Demand. And this is how most of us at MSI access our supercomputer. So it is highly recommended. You get there by going to this link right here, ood.msi.umn.edu, and you'll see a window something like this. So here's mine right here. Mine looks a little bit different than yours because I'm a member of many different groups, but presumably if you logged in, it should show something along these lines. And just very briefly, the navigation bars at the top let you access a lot of very useful things. The files will get you to the files in your group as well as in your home directory. Jobs is where you can see any of the jobs that you've got currently running. This clusters dropdown will let you access the command line terminal directly. And then really usefully, I use it all the time under the interactive apps. We have a number of applications which you can run on the supercomputer and use to access and analyze your files which are on the supercomputer. So I almost daily have an RStudio server interactive app open, oftentimes an IGV app too, to see where my mapped reads are in different genomes. All right, I'm going to be switching back and forth between the tutorial because otherwise I'm going to talk myself into a corner. Okay, yeah. All right, so the first thing that we are going to do, is we are going to look at the input for this experiment that we are going to analyze, this tutorial data. So we're going to do that by looking at the reads on the command line. We're going to click on the clusters tab right here, and then click on agate shell axis below there, which should open up a new window, which looks roughly like this. Yours is not going to look exactly like mine because I've modified what loads whenever I open up a terminal window, such that this information right here is not going to appear on your screen. What you see down here at the bottom is what's called a prompt. On the left-hand side of this percent symbol, you've got your login name, and then it tells you what node you're actually accessing on the supercomputer. And then within these brackets, it tells you what directory you are at within the supercomputer, and a tilde refers to your home directory. And then on the right-hand side of the percent sign is where you actually type stuff like that. If I type some gibberish and hit enter, it's going to tell me command not found. We're going to change directory and go to a place called common, where us analysts at MSI are able to put useful materials for tutorials. To change a directory, you type in the word C or the command cd or change directory, and then hit space. And then importantly, since the common directory is at the top of the directory tree, we're going to start with the /. I'm going to type in common. And if I start typing a few letters and then hit tab, once I've hit a unique directory, it'll finish it for me. Then I'm going to hit B for bioref, which stands for our biological references. And then I'm going to hit enter there because I don't remember exactly where we are at that point. So now you see that the prompt has changed to show our directory is now common bioref. And let's see what's in here. That'll help us navigate to the tutorials. And to see what's in a directory, we're going to type ls and then hit enter. And we see a lot of different things right here. Am I in the wrong spot? And I realize now that I'm in the wrong spot, so I'm going to actually go up a directory by doing cd space dot dot. Now you see that we've gone to common. We're no longer in bioref. I'm going to look where we are again. That's where I want it to be. And then there's the tutorials directory that I'm going to go in. So I'm going to now hit cd space tutorials, a tab completed, hit enter. I'm going to look in there. We are doing a bioinformatics tutorial, so we go in there. We'll look in here. We're going to do the bulk RNA Seq tutorial. We're just going to keep doing this process of looking and navigating. And then finally, the reads, the FASTQ files for this tutorial are in the reads directory, so we're going to go in there. Now when we ls, we see 16 different files. So let me briefly show you bit about this experiment. So this particular experiment is from an experiment on mouse in which they were studying whether there is differential expression in mononuclear blood cells from bone marrow or from spleen. And they had four unique individuals per treatment, so a grand sample size of 8 mice. And since these data were sequenced in paired end, and we end up with 2 files per each sample, we end up with 16 files total. And that's why we see 16 files here. We also see that they're named based upon their treatment and based upon their replicate. We can also see if we look in one pair, for example, if we look at the bone marrow, this first one right here, we see that the first bit of information is identical for the first 2 files. It's because they refer to the same sample. And then really the only thing is different is that one is ends in R1.001.fastq.gz and one ends in R2.001.fastq.gz, indicating the first read and the second read per each fragment. Sorry if I'm making you scroll sick. Oh, yeah. Good, that's what that should do. So let's take a look at one of these files to actually see what the reads look like when they come off of the sequencing machine. We can't actually look directly at one of these files because you see that they don't just end in .fastq, which is a raw text file. They end in .fastq.gz. It stands for gzipped. It's a compressed file format. And so we are not going to be able to look at that directly. If I try to look at that using this command called head, which would show you by default the top 10 lines in a file, and then type in the file and hit enter, because it's a compressed file, we should not see readable text. We should see a bunch of gibberish, and we do. So in order to actually see what's in that file, we're going to decompress it sort of temporarily. And to do that, we're going to use this command called zcat. That will decompress the bits that we need to use. So I'm going to type in zcat. Here, actually, I'm going to show the files again so we see which one we're doing. Zcat and then space, and then the name of that file. And I'm not going to hit enter yet, because if I hit enter right now, it would decompress the whole file, and we'd see everything stream down our system. It would take a while, and it would be hard to parse. So what we're going to say is we're going to take this decompressed bit, and we're going to pipe it into that head command so that we can just look at the top, in this case, let's say 8 lines. And to pipe something from the left to the right, you use this vertical bar, which is usually above the it's the shift key and then the backslash, usually right above your enter. And then that means in Bash that anything on the left will go into the thing that you type on the right. It's really useful. Oftentimes you'll pipe multiple things into more and more programs to make modifications as you go. Similar, if any of you use R, to how you would use piping with dplyr or magrittr. So we're going to pipe that into head. And I know, because I know how FASTQ files work, that in FASTQ files, there's 4 lines for each read. And so I just want to look at 2 reads worth of information. So I'm going to look at 8 lines. And to tell head that, I'm going to do -8. And so now hopefully this should decompress just the top 8 lines and show them to us. And so that's what we see. So this is what the data look like when they come off of the sequencing machine and they're sent to you in a FASTQ file. There's 4 lines per read, and the first line for each read always starts with this @ symbol. So you can see this @ sign repeat down here. The first line is metadata, which tells you what read this is. It's basically the read name. There's a bit more to it than that, but we're not going to bother with that. That's really all you need to know about it. The second line, which is continued on to the third line, and I recognize it's a little bit confusing, but this is all one line right here. is the actual sequence that the sequencing machine predicted based upon its read of this fragment. The third line is some metadata. It always begins with the plus, and oftentimes there's nothing after the plus, depending on what sequencing machine you're looking at. And finally, the fourth line per read is the quality score. So each of these symbols here represents a different Fred 33 quality. So you can look up a Fred 33 quality table if you want to know what all these letters and symbols mean. But these are letters are generally on the higher side of the quality scale. And then we see a similar sort of thing for the second read in this R1 file. And one thing that's nice to note, just to have in your mind, is that these read names, so the first line per each read, should match a read name in the R2 file, since each fragment was read 2 times, usually from left to right and from right to left. So let's repeat this looking at the top, but not now. Let's swap out the R1 for the R2 to look at the other read for this one, and we should hopefully see a very similar name for the read, with the exception that this /1 should be replaced with the /2 to indicate that it's the second read per this fragment. So I'm going to press up, which shows me my last command. And I'm going to press left a bunch of times to get over to this R2, change the 1 to 2, hit enter. And that command was put in right here. And we see exactly what we predicted. So here is the name for the first read of this fragment. Here's that same name, but just ending in /2 and /1. So this is the read, this and this are the read pair for the first fragment for that was sequenced in that sample. So that's what the FASTQ files look like. My video commands keep showing back up. That is so annoying, Zoom. A fun little gut check that we can do is that since this R1 and this R2 file sequence the same fragments, we should have the same number of lines in both of these files. And we can check that with the command WC, which stands for word count. And if we give it the argument -L, says how many lines are in this file. So now, instead of piping in the decompressed file into head, I'm going to pipe it into word count, WC space -L. It's going to read through the whole thing. It's going to take a second to count how many lines there are.
It's a question that chat.
You want to just read it out?
Looks like the quality in the second read file is lower. Is that common?
Yeah. So a question from the chat was, is it common to see, so someone observed that there's more lower quality symbols in the second read than in the first read. And depending on the sequence technology, that is often very expected. Good observation. And nothing to worry about, too. So we see that in the first file, we have 4 million or so reads. We should have the exact same number in the second file, unless something got corrupted between the sequencing company and us getting the files. But in this case, we see the exact same number of reads. So that's a nice little quality control. There's some really important information that we care about from a bulk RNA-seq perspective that is missing from these FASTQ files. It, of course, doesn't tell us what gene they're in. It doesn't even tell us where in any organism these reads map to. And so that's why we need to do a sequence analysis to figure that information out. So the first graphic that I showed you was a really high bird's eye view of getting from the experiment all the way to the analysis. This graphic right here focuses on the sequence analysis, starting from those FASTQ files that we've now explored in a little bit, and ending in the counts matrix, which is the starting point for all of the statistics. Our chirp pipeline, and any other pipeline that you might find that would do something similar to this, breaks this into 2 main steps, because the first steps can be done on each sample independently, there's no crosstalk between samples. And then the downstream steps, where we're putting it together into a experiment-wide counts matrix, does need to pull in information from all of the different samples. And so in our pipeline, this is broke into two different pipeline files. This is more information than you'll really need to know, but that's how it happens under the hood. And what gets done on every single file independently are these steps right here. First, we do some trimming and some quality control. We run the classic piece of software called FastQC to get statistical information on our read quality and length and number. And we trim off adapters, which may have been added for the sequencing process. And we also trim off low quality parts of reads or even whole reads themselves using a piece of software called Trim-O-Matic. Once the trimming has been done, then we need to figure out where these fragments are in the genome that we expect them to be in. And so to do that, we're going to need to provide another piece of critical information, which is the genome that we are going to map to. And an important nuance here is that each piece of software that maps your reads to a genome has its own flavor of genome that it wants. So while all of these softwares start with the genome in what's called a FASTA format, which is just the sequence of each chromosome, they all want a specific type of index file. In our pipeline, we use a mapping software called HiSat2. We chose that one specifically because it's what's called splice aware. As anyone that has taken a genetics course knows, if you are dealing with eukaryotes, their genes are split into multiple exons interspersed by introns. And so the RNA, which gets sequenced, could go from one exon to the next exon and skip that intron. If we mapped naively to a genome without knowing that, it's going to think there's something weird because there's this big chunk between the left-hand side of the read and the right-hand side of the read. And so we need the mapping software to be aware of the splicing that gets done. And HiSat2 and then also STAR are two of the main pieces of software that do splice-aware mapping. So we'll need to provide our genome in HiSat2's flavor of index. Once that is run, we'll have a file called a BAM file. And that file will tell us for each read that we provided it in the FASTQ files, where in the genome did it best match to, and how well did it match to it. Once we have those mapped BAM files, we need to do some sort of housekeeping on them, really just to get them in the right order, because different software expect these things in the right order, and make it indexed so it can be searched through faster. That's pretty easy. And then we end up with these sorted, indexed, mapped BAM files. And these files, they tell us where in the genome each of our reads are. But they have no idea what's a gene and what's not a gene. They just know it went to this location, position 1,433 on chromosome 1. They don't know if that's in a gene or not. So then we need to provide yet another piece of information which says where in the genome are all of the different genes. And that kind of file is a GTF file, somewhat interchangeable, but not perfectly with a GFF file. And this file, in contrast to being the sequence, which is in the genome FASTA, or the index, again, tells us where all of those exons are, which genes they belong to, etc. And so then, by comparing that file to our mappings in the BAM file, software called feature counts can count up how many reads were in each exon, sum that to each gene, and then once we've got that, do it for all of the samples and smush all of those gene counts together into our counts matrix. Cool. So At that point, we've got the counts matrix. And what happens at that point is that R gets run on our counts matrix to do the analysis for us. It takes in the raw counts matrix, which I'll describe what that file is called in a bit more detail later. It normalizes it so that those differences in depth are not obscuring biological differences in our results. And then it does both unsupervised analyses to create hierarchical clustered heat maps of our data and principal components analysis. And if we told Chirp about what group our samples belonged to and whether or not we had a statistical comparison that we wanted to make, if we did that, it will also compute differential expression statistics. We'll go through that a bit more later on. So we are going to now look through this step a bit more, figure out what information we need to give to Chirp in order to get it to run. We know now that in addition to our FASTQ files, we're going to need to give it a genome in HiSat2 index form, and we're going to need to give it a GTF so that it can count where all of our reads are in the genome per gene. All right. On MSI, if you haven't used MSI before, one thing that we provide to everybody is extra space, which is temporary. It's really fast space, so it will read and write really quickly compared to the more typical group space. And you get access to a large amount of it, but it's all deleted after 30 days, no matter what. So it's really good for temporary things that are going to produce a lot of big files. Maybe you only care about a couple of those. So maybe then when your software is done running, you copy your needed files to your actual project space and stop using what is called the scratch space. We're going to go to that scratch space because that's where we're going to put our output from this pipeline. So to do that, we're going to type in cd space for change directory again. And this one is called /scratch.global. If this is your first time doing a tutorial or using the supercomputer like this, you may not have made a directory for yourself in this scratch space. A useful variable that is included in our version of the supercomputer is the user variable. Variables in general on the command line are prefixed with the $sign and then the name of the variable. There's one command called echo, which says, tell me what's in the thing to the right. So I'm going to type in echo, and I'm going to hit space, then I'm going to type in that $sign user. And when I hit enter, it should say CHACO001 for my username. And for all of you, it should say whatever your X500 or MSI login is. So that's what it does say for me. It says something different for you. We're going to use this user variable so we can all be typing in more or less the same thing. And I have already made a subdirectory with my username in it. So if I do cd space and then type in that variable again, hit enter, it's going to go into my directory. If this works for you, great. If that doesn't work for you, and you get an error saying that that directory does not exist, then what you can do is you can make it. You can do mkdir for make directory space, and then type in your user again. This is actually going to give me an error because my directory already exists, but it should work for those of you who don't yet have this directory. Now, once you have made that directory, let's go into it. I use my Scratch directory, so I have multiple things in there, which I can see, again, using that ls command to list this stuff in there. You all, if this is your first time doing it, should have nothing in there, or maybe you are already accessing this. We are going to make a directory to hold our output in there. I believe we're going to just call it tutorials. Let's check so I don't do it differently than the No, we're just going to leave it like that. Okay. We're just going to sit here for a second. To access software that we've previously installed on MSI, we use what's called the module system. This is something that's been implemented that is accessible on many different supercomputers if they're using what's called Slurm, which is what we use to submit jobs. Type in just module. And you can find out about software that's available on the supercomputer either by going to our website, or if you know the name of something that you're interested in, you can type in avail and then type part of the name of the software and see what pops up. So for example, I know that we're going to use HiSat2 inside the pipeline. I'm not going to access it directly. Let's just check and see if it's available. I'm going to type in HiSat. And I'm going to hit HiSat and then star because I don't know exactly how they wanted to spell it. If they wanted the two or if that's just in the version. And I'm going to hit enter. And we see that there is some software for HiSat2 available. The one that we're actually going to use, our pipeline, is called Chirp. So we're going to repeat this process and see which versions of Chirp we've got installed on the supercomputer. So we type in module avail Chirp, all lowercase, and I'm going to hit enter. I'm not going to bother with the star because I know by heart that it's just called Chirp. What we see here is that there's multiple different versions of Chirp available. In almost all cases, unless you're doing something very specific and you know exactly why you're doing it, you should likely use the latest version of a piece of software. You'll always want to ideally load the same version within a project as well. So say you started this project today, you loaded the Chirp version 1.1.0. We made 1.2.0 before the project was finished. We would recommend you keep using 1.1.0 until you published or whatever. But when you're starting a new project, you should probably always start with the latest version of the software. To load software with the module system, you type in module space load, and then the name of the software. We're going to type in chirp slash 1.1.0, hit enter. And it gave us some information. This suggests that it successfully loaded itself into our system. It says when it was last changed, shows how to see what the latest changes were. And then importantly, it tells us that chirp can be called by running $chirp. So that's a kind of a fun quirk of Bash is that variables, and we know this is a variable because it starts with a $, can even hold directions to starting a piece of software. So if we typed that in and hit enter, we should see some message from Chirp. And we do. It's a bit, I realize now we should probably update some of this messaging a little bit, but it says that to use Chirp, we type in Chirp like that, and then use some sort of subcommand and possibly some options. And it tells us that there are 3 subcommands which we can use here. Bulk RNA Seq, which we're definitely going to use, group templates, and then genome aliases. We're going to mostly focus on bulk RNA Seq. Let me make sure I'm not getting too far ahead of myself. No. And multiple of these subcommands have help, where if you type in -- help after the command, you'll get help information. That's common for many different Bash software to be able to access help like that. Let's type in chirp, then the name of the subcommand, bulk RNA Seq, and then space -- help. And then hit enter. It'll give you the complete help. I'm gonna have to scroll up so we can see the top of it. And what we see here is a list of all of the possible commands that you can give it at the very top, as summarized as possible. And then just below that, it tells us the required arguments, and that's what we're going to focus on first. And then finally, a lot of optional arguments. We are not going to go through all of these optional arguments in this tutorial, but it could be useful to look at them and see which may be meaningful to your experiment. But when we look at these required arguments, let's focus especially on the top 3, because the 4th one is a bit of a surprise. We see that we require this one, the FQ folder, So we would type in --fq folder and then some folder directory, and that's got to be the directory that contains FASTQ files. Unsurprisingly, we know that we also need to provide it with the HiSat2 index and a GTF. One of these we already know. Does anyone, either online or in person, know which of these we already know to be able to run our tutorial experiment through Chirp? Anyone online? Oop, got some chats. FASTQ directory. You're exactly correct. And so earlier, when we were looking at the FASTQ files, they were in the directory where that we will have to supply as the first argument. So when you run chirp, all of your FASTQ files must be in one directory, and it's that directory path that you'll provide. So this is the command that we're going to be building up from scratch. To submit this job, we're going to do chirp bulk RNA Seq, and then we're going to have to tell it the FASTQ folder, the HiSat2 index, and the GTF. And just like Anu told us in the chat, we already know the FASTQ folder's path. We navigated to it before, and it's this: commons/tutorials/bioinformatics/bulk RNA-seq/reads. We don't yet know where the HiSat2 index that we need are or where the GTF are. For this tutorial, and for most users' actual experiments, we have provided a database of reference files for everything which is in the Ensemble releases. This does not include, and I apologize to those cyanobacteria folks, prokaryotes. For those, you'll have to find your own genome and GTF and index the HiSat2 index yourself. There is information on how to index your own genome file for HiSat2 on our system at the end of this table of contents. And this is the sort of thing we are always happy to help with if you send us a help desk ticket as well. So we are going to go and find where the HiSat2 index are and where the GTF file are for our organism, which is the mouse, Mus musculus. We're going to go and find it in this path, which is where we host our bioref database. So I'm going to go back to the command line. And I'm going to change directory again, and I'm going to go to common/bioref/ensemble and enter. And I'm going to look in here with ls. We see a number of things, most of which you can safely ignore. We have some old updates from previous years in these directories, which contain sort of more classic information that you don't need to worry about. And then in the directory named by their taxon is where you're going to find the genome information for your species of interest. Whereas most of them are taxon named, all of the model organisms all are in the main folder. This is Ensemble's decision. They're model organism biased. So we're going to go into main. And if I hit ls in here, I should see a large number of directories. And they're named, as you can see, by their species name. We also contain 2 different releases per each organism. If that doesn't mean a lot to you, don't worry about it. For this one, we're going to use 113 because it's what was written when the tutorial was written. These don't change that rapidly. It doesn't actually matter that much which one we use. But I know that mouse's name is Mus musculus. So I'm going to type in mouse, hit tab, and see, oh, I've got all these different options. So I'm going to have to keep typing before it'll let me tab complete. Type in an underscore, musculus. I hit tab a couple times. Up, still some more options. We're going to just do the main model species in the 113 release. We'll type in -113 and then hit enter. Oop. What did I do wrong there? Don't know what I did wrong. Okay. Now when we're in here, it's going to be kind of a pointless directory. So we're going to go into that as well. Go in here. And now we're starting to see some interesting, useful stuff. We see 8 different directories that each contain different pieces of information about this genome. This directory structure is repeated in each of our different organisms in the Ensemble BioRef database. The basic sequence from which everything was derived, so the plain text FASTA file, is in this Seq folder here. So if I go in Seq, enter, and look in there with ls, I see a number of things. I see this done file and this checksum with wget_log, remote checksums. This is all just information that told us during the creation of the database that things went as they expected. But what we really care about is this one, mus musculus, blah, 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 .fa, which stands for FASTA. This one, which is genome.fa, and this one, which is .fa.gz. So these files, these .fa, should contain the actual genome sequence of our organism. So we can look at the head of it, because it should be a plain text file, and see some sequence information. And at the beginnings of chromosomes, there's usually not great sequencing information, so it's actually going to look kind of junky. That's okay. We see that it starts with chromosome 1, and it doesn't actually know what the first bunch of bases are, because we don't know it's at the telomeres in mouse just yet. Genome.fa, we've provided in each of these directories, which it's what's called a soft link. It points at the full name file for that organism. And that just provides a nice little shortcut, so you don't always have to remember exactly what this file name is. You can just always use the Seq folder /genome.fa, and that will be the same information. So if I do head genome.fa, it should tell us the same thing. Great. This is not what we're going to provide to CHIRP, because this is an unindexed genome at the moment, and we know that we need to provide to CHIRP the HiSat2 index version of this genome. So I'm going to go up a directory, cd space ... I'm going to look again with ls, and now I see that there's another directory here called HiSat2. I'm going to go in there, cd space, I hit H and then tab complete it, ls to look in there. And here is where we see the critical piece of information for our second required argument to CHIRP. We see these files, which are all very, very similar, except that they end in different .1, .ht2, .2.ht2, et cetera, et cetera. And this group of files comprises HiSat2's index for this genome. And what we provide to CHIRP as the HiSat2 index needs to be the complete path, directory path, to the index, and the prefix, which is shared by all of the index files. So when we provide this to CHIRP, we'll have to write -- HiSat2 - index space this whole path / genome. I'll show you that right here. Just like this. And then that will tell CHIRP how to access that HiSat2 genome. If you're doing it on a different species, parts of this will be slightly different, but otherwise it'll look similar. The final critical piece of information is the GTF file, which is what feature counts will use to count the reads per gene, counting from the BAM file, which we get after mapping with HiSat2. We also provide those GTFs, and since the GTF annotates where in the genome the different genes and exons, et cetera, are, it'll be in a folder called annotations, or annotation, excuse me. So I went up a directory again with the cd ... looked at our group of directories. I'm going to go into the annotation one, go to ls to look in there again, and we see one that ends in .gtf right here. And we can look at the top of it with head, TF. And since there is an uncompressed version here, we're going to leave off the .gz so we don't have to bother with that whole zcat process that we did with the reads, the FASTQ reads earlier. And we see something that looks like this. We see some comments, and then we see a row for each feature in the genome. This particular feature is a gene. It says what position it starts at, what position it ends at. It gives it an ID, etc. I think I have in this tutorial a little bit of information on what's in all of those. I guess. These are the names of the columns in a GTF. There's also this link to the official GTF documentation if you want to learn more about them. But basically, there's sort of a hierarchical structure where it shows each exon, which belongs to each transcript, which belongs to each gene, which belongs to each chromosome, which belongs to this organism. And that's what is used to count features. So now we know that our path for the GTF is this path right here, ending in musmusculus.grcm39.113.gtf. So our command right here, so far with the required pieces of information, looks like this. We could, in theory, run this just like this, but we have not given any information as to what different treatments our FASTQ files belong to. So it won't know whether it should do any differential expression or anything like that. And we also haven't given a couple of critical pieces of information related to how these kind of old data were sequenced, which we'll talk about a little bit more in a minute or two. But first we wanted to show a little helper task right here. And if you recall, when we looked at the help for chirp by typing in $chirp space bulk RNA seq space -- help, and I can just do that again for you just to remember. Scroll up past all that optional stuff to the required things. The fourth required one was -- organism. And it says what that is an organism shorthand for automatically setting a HiSat2 index and GTF annotation file. And so it's a little bit of a misnomer because it's under required, but really you can either provide the full path to the HiSat2 index and the GTF, or you can use our little helper -- organism. And so what that is for the most commonly used model species here on campus, you can just type in the organism's name. And to know what the organism is, we've got that other subcommand for chirp. When we typed in chirp, we saw genome aliases. And so if we run that by typing in chirp space genome aliases, it tells us if we're studying any of these organisms right here, instead of finding its HiSat2 index and its GTF file, we can simply use the -- organism command and then type in the alias right over here. So for us, it will be mouse. And so what we've got here is that these two commands are identical in how they function. Here, we're being more explicit about saying where the HiSat2 index and the GTF are. Here, we're using the -- organism mouse genome alias method. We're doing pretty good on time. So here is that same graphic that we looked at before, but now just showing a little bit of what's in each of these files. To make this run, we're going to provide our FASTQ files, which are the raw read information from the sequencing machine, one pair per sample. We're going to provide the HiSat2 index version of the genome. We're going to provide the genome annotation, which says where all the exons are in that genome. And then those things are going to allow us to map the FASTQ reads to the genome in the first step using HiSat2. And then taking those mappings with the BAM from the BAM file, messing with them a little bit, and then once they're messed with, comparing them to the GTF file to be able to count using feature counts how many reads were in each gene. Okay. So, as I mentioned earlier, we have not told Chirp anything about the experimental groups. And like I said a second ago, in theory, we could just run it. It would still produce our counts matrix. And then we could do our analysis completely ourselves if we wanted to, starting from the counts matrix. But since Chirp does have functionality to make the unsupervised graphs, to do some simple treatment versus control differential expression, we're going to use that functionality. So we're going to start by making an output directory where all this data is going to go. Let's go back to our scratch.global directory that we made. We're going to make a subdirectory called RNA Seq tutorial. So I'm going to do that as well. Let's cd space scratch global user. Now, when I hit enter, you can see that my directory changed from where we were in bioref to where we now are in my directory. Yours should say your X500 instead. We're going to make a directory using mkdir, make dir. And I'm going to look at that again to make sure that I don't have the capitalization incorrect. I'm just going to copy and paste it. And as a reminder, this is still also over here in the cheat sheet. Right now we're around here. And after I make that directory, when I hit ls, I should see it in there. There it is, RNA Seq tutorial. Okay. The way that we currently provide information on what groups samples belong to Chirp and what contrasts we want to do between those sample groups is by providing an Excel spreadsheet. That's in the process of being augmented to also be able to provide just a CSV file. But I like the Excel spreadsheet file because it kind of keeps the group information and the contrast information all in one spot. And so what's also nice is that we can make one of these files to fill in ourselves using Chirp, and that way we can be sure that Chirp's finding the right samples that we need to provide a group name for. And if you recall, one of the subcommands that Chirp has, in addition to bulk RNA-seq and genome aliases, both of which we've looked at, was one called group template. And if we want to provide a group template for bulk RNA-seq, we will do a command similar to that. And here, let's look at the help for that and see what it tells us about that. So I'm going to go back to the command line, type in chirp, group, template, bulk, RNA-seq, space dash dash help. And for this, there is different information required. All that is required is your FASTQ folder, which is the same that you'll be providing later on. That's the one that's common tutorials or something like that, common bioinformatics. We'll find it in a second. Some optional arguments. The only one that we're going to use is output. By default, it's going to save the group template output just in your scratch.global/user directory, but we're going to put it into this new directory that we just made. So, right, this is the required input, same as the one that we're using for the main chirp command. And we're going to have the output go into that directory that we just made We're going to call it tutorial RNA Seq groups.xlsx. And this time I am going to copy paste because I do not feel like typing all that. And I would like to point out one other cool thing about Bash when I do that. You see right here that I have not put this command in all in one line. That would be very long and hard to read. Instead, I have spread it between multiple lines. And the way that I've spread it is with a forward slash. Forward slash, they are interpreted by Bash as if the stuff on the next line is just a continuation of the current line. And so that's a really nice way to make things readable to a human without messing up Bash. I'm actually going to copy it from the cheat sheet because I believe I also noticed, maybe I didn't. I thought I saw, there's a gotcha with these backslashes. If there's ever anything, even a space after the backslash, it will not know what to do. It's going to give you an error. You're going to be like, what the heck is going on? And it's going to be really annoying. So whenever you're using these backslashes, make sure that there's nothing like this accidental space right here after it, or else it's going to screw you up. So I'm going to go to the cheat sheet, which is right here. And here's the command that I'm going to use. This one might also have space. We'll see what happens.
Jeremy, could
Yes.
you also put that in the chat? I wasn't able to
Absolutely, I'll put it
log
in the
into that.
chat. Okay, I guess it didn't copy the spaces. That's good. We'll see how this copy pastes from the chat to your terminal. Yeah, if you need any more help, I'm sure that Alyssa or Juan could help grab that for you. Okay. So I ran that, copied and pasted it in here, hit enter, said a thank you message. Importantly, it said success. And then it said template file is here. It said all the sample groups are null. Please edit it, blah, 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 before you submit. Now we're going to use Open on Demand again to access our files a different way. Sometimes you're going to be navigating the command line. Sometimes you're just going to be navigating with the drop-down menu. I'm often doing a combination of both, navigating when I'm running stuff, and then when I'm wanting to look at plots or HTML output, I'll do that through Open on Demand. So we know that that file was put into our Scratch folder. So I'm going to go to the files. Yours is not going to have this many because you're not a member of many different groups. You're probably just a member of one group, one or two groups. But at the bottom, you should probably have scratch.global, possibly even one that says scratch.global/your username. If you don't have this, here, you know what? Let's just all do this together to make sure that we're in the same place. Just click on the scratch.global one. This is going to take a little bit of time because the browser needs to read all of the folders here in scratch.global, which is any folder that any user could have possibly made. But when it's done thinking, we can use this filter over here to find our username. And hopefully this just, okay, cool. Hopefully yours isn't taking much longer than that. So I'm going to come over to this filter and I'm going to type in my username. You'll have to type in your username. And you could type in mine, you'll see it actually, because it's in the scratch.global folder, which is visible to everybody, but you won't be able to get into it. You'll only be able to get into yours. Click on it. When I click in it, now I'm in my folder, which we can see by this directory information up here. And then we see all of the stuff that I've got down here. And we see here's this directory that we just made, RNA Seq tutorial. We see that we just made it because it says the time that it's stamped over there. But clicking there, we do see that XLSX groups template file, which we also just made. We're going to download it by clicking on the dropdown, hitting download. Then we go to our downloads. It's obviously the third time I've given this tutorial with this file. Open it. Hopefully you have open it up in numbers. Do that. You're going to want to open it up in something like Open Office, regular old Office, something that can open up an Excel spreadsheet and edit it. Not numbers. Kind of annoying. Okay, it's a little bit bigger for everybody. So what's really useful is that we see that it did indeed find those 8 different samples, and it did that by looking through that FASTQ folder which we provided, knowing that r1_001.fastq.gz is the expected suffix for each of these files, so everything before that must be the sample name. And then it gave us null for all of these groups. There's also a second tab right here at the bottom called contrasts. So we're in the groups tab. There's also the contrasts tab. And what we're going to do first is assign the samples to a group. Now we have 4 replicates of bone marrow and 4 replicates of spleen. And sort of logically, we can just call these ones bone or bone marrow, whatever you feel like. They should be all the same because all of these samples belong to this group. We'll make up a different group for spleen. I'm just going to call them spleen. Just like that. And we know from this experiment that we want to compare expression in the bone to the spleen. And to tell it that we want that statistical contrast, we're going to put that information in the contrasts tab. Let me look back at my tutorial real quick, make sure I get the denominator correct on this. And the way that we're going to set this particular one up is that we want, when we see the information, the metric that refers to the difference in expression between one group and the other, that's called the log fold change. When it's higher, we want to know that means that it's more highly expressed in the bone marrow. And when it's negative, we want to know that it's more highly expressed in the spleen. And so to do that, we're going to set the test group will be the numerator group, and the reference group will be the denominator group. So I'm going to come back over here. The numerator will be test group. Reference group will be the denominator. And critically, these group names must match identically group names that you've put in the first sheet. Finally, one should give it a comparison name. This can be really anything. I like to give them just in terms of the numerator underscore versus underscore the denominator. That is just my shorthand for always knowing which was in which spot. So I'm going to go bone versus spleen. A couple of gotchas here. Please do not start any of these group names or comparison names with numbers. Please do not use spaces or special characters because those may break chirp. We have tried harder and harder to catch as many of these edge cases as possible, but we have not caught all of them yet. Okay, so you save this once you've got that all done. Oops. Save it as whatever you want to call it. No, call it. Yeah, sure. Yes, I'm going to replace it. I know. Oop. Make sure you save it as XLSX. Open on demand or Open Office wants to save it as or even just XLS is fine too. Close it up. Then you'd upload it back here by just coming back to Open on Demand and clicking Upload, browsing the files, and finding it on your system. And double-clicking and hit Upload. I would like to also point out that if you're having any trouble with getting this to work, maybe you don't have OpenOffice on your system, we also have made a filled one for you and put it at this path right here. So if you can't get through this step right now yourself, you can just use this path right here. So once you've got that filled in and uploaded, that piece of information, the path to that file, will be what you give to this fourth argument here, dash dash expr dash groups for expression groups. So now if we ran this, it will know which groups our samples belong to, and that we want to do one statistical contrast of the bone versus the spleen. All right. So now this is the command we're actually going to use to submit. And surprise, there's a few extra arguments here, which we have not talked about. So one of you copy this into the chat. Thanks. So
I don't.
I'm going to just go through this briefly. We know the required argument of the FASTQ folder. We know that we can use the shorthand, the organism flag, say mouse. We know that we're going to give it our groups information with this Excel spreadsheet. And then after that, there's a handful of optional commands which we're going to use. We made this directory, RNA Seq tutorial, so we might as well use it to have our output collected in there. So we're going to use the optional argument dash dash output dir, and that's going to say that our actual output is going to go into the directory that we supplied to it. And for us, that'll be scratch.global, our username, that RNA Seq tutorial directory that we just made, and then a subdirectory called out. We're also going to put all the working temporary files into a similarly named file in case we want to look at them later. It's going to end in work. This one is critical for this data set specifically. It's probably not critical for your data sets. This particular set of sequence files was sequenced with an NEB Ultra, I think is the name of the kit, which creates unstranded paired end RNA Seq libraries, FASTQ files. So we need to tell it that it's dash dash strand U. Most things these days are the reverse read first, then the forward read second. In other words, RF, which is the default strandedness in chirp, which would be dash dash strand RF. Occasionally there's forwards and then reverse, in which case you do dash dash strand FR. If you have questions about that, email us and we'll help you. And similarly, whatever company or core sequenced your data should also be able to tell you that information. Because we want this to run quickly, we're going to use the MSI queue called MSI small. This has access to the largest number of nodes on the supercomputer, but they don't have the largest amount of compute resources on each of those nodes. Doesn't matter for this case. I actually almost always use MSI small, even with my actual research. We're going to tell it that we're going to give it 8 processors per node. That will let some of the steps of the pipeline work in parallel with up to 8 processors. For a full-sized RNA-seq project, you might use 24. We're going to tell it that it should have 24,000 megabytes of mem or RAM in each of these steps. That's on the low-ish side, but it is just fine for this. You could try, 36 or 48,000 and see if that works for yours, and then bump it up a little bit if necessary. We're going to give it a maximum amount of time of 2 hours with this dash dash wall time argument. Again, this is set low on purpose so that this runs quickly during the tutorial. You might want to bump that to 8. That's probably going to be good unless you're working on very large files with a large genome. And then finally, we are not actually going to submit the job when we submit this command. We're going to do all the preparations and then we'll submit it in the second step. So you can copy this whole thing, either from here or from the cheat sheet. Go back to the terminal, paste it, hit enter. Hopefully it won't give us any error. I'm gonna scroll up to where I submitted it. You see where I submitted it, ending in that no submit. And what it says is it gives warnings. Typically in bioinformatics software across the board, warnings should be looked at, but not be considered serious cause for concern. Errors are what are serious cause for concern. And they're just warning us that these directories did not exist, so they made them for us. Then it said success. And what it meant by success is that the sample sheet and pipeline script were created, and it tells you where those pieces of information are. And what it says to do, it tells you how to actually finish running it, or finish starting the run. After you verify the information in here, you can run it by just typing in bash and then giving this full path, and yours is going to be slightly different. Mine says .chaco001 because that's the dollar user I gave it. Yours would probably say yours. Into the terminal, and then this will actually submit the job. Let me look at my tutorial, make sure I'm not missing anything. Okay, yep, and here's in the tutorial, it's got the explanations of those additional arguments. Okay. Right. So when we see this information that says that these scripts were made, we see that they were made in this particular location, scratch.global, our username, RNA Seq tutorial, out, and then the name of the files. So let's take a look at those. We could do that through Open on Demand, the file browser. Since I'm already pretty close by in this terminal, I'm just going to do it here. We'll go to RNA Seq tutorial, out, hit enter. I hit ls, I should see those. I do. See 3 files. These 3 files each have 3 different pieces of information. This one that ends in .pipeline.sh actually has the pipeline put together from our pipeline and your input information. And you'll never really have to look into that. But if you're intrigued in extending your bioinformatics journey, that could be a useful file to sit down with some tea and look at to see how the things are actually being run. This one that ends in Q sub array, that you should never really have to look at. We might look at it during troubleshooting if anything goes wrong, but that's just going to associate each of your samples with a supercomputer MSI submitted job number. But the one that we are going to look at briefly is this one called the sample sheet. That file tells Chirp which FASTQ files are associated with which groups and what genome index and GTF should be used to analyze them. And I'm going to look at it with good old head as usual. And I'm going to type in head 2020 blah blah blah, hit tab, and then we can type in sample sheet, hit enter. And in this case, we see something like this. We should see for this tutorial data set, 8 different lines. So we could check that actually. But instead of typing in head, we can do word count dash line. Oh, both. Oh, I think that's because no, it says line. But whatever. Ignore me. We're gonna press on ahead. We'll look at back this again. The head. I'll figure out this weird word count thing later. But when I look at the sample sheet, I see the first piece of information is a single sample's name pulled out of that FASTQ file. And then after a pipe symbol, the group that it belongs to. And then after the next pipe symbol, the actual FASTQ file read 1, then the FASTQ file read 2, then where it should go when it is output, and then some information on some of the basic trimming data that we are going to use, where the HiSat genome is, where the GTF is, etc. So when I look at this, what I check is, does my sample name, is it correctly associated with my group name? And is it correctly associated with these FASTQ files? I'll check a few of them, especially if I've got some 150 sample experiment that I'm doing. And if it's looking good, then we can press ahead. In this case, it's looking good. And we will, yeah. I don't know why it says, it's got 12 there. Whatever, let's see what happens. That command from before said that we can type in bash space and then the full path to this pipeline.sh. But since we're already in this directory, we can omit the path before the file. So we can just type in to actually submit the job bash space and then the one that ends in pipeline.sh. Cool. And if this worked for you, you should see something similar, but a bit different to what I see here. It should say that it's setting an account to your account. That's going to be something like tutorial or your lab's group name, the version of Chirp that we're running, where the data is going to go, where emails are going to be sent, and then some information on the job array ID and the job ID. This is saying where in our supercomputer what their job ID names are. Yeah, cool. To check on the status of your job, Slurm, which is what controls jobs on our supercomputer, has a command called SQ. The letter S and then Q as in standing in line, QUE, QUE. And then you type in -U to specify a particular user. And then you can use that $user to specify yourself and enter. And in my case, I see, salute, that I already have 8 jobs in the running state right here. And the names of those jobs are prefixed with bulk RNA. So these are those single sample jobs going where for each of our samples, we're going through the trimming, we're going through the mapping, and then the sorting and indexing of the BAM. That's happening right now for each of the samples independently. And then this final one that's in the state PD for pending, this is going to wait. We know it's waiting. We know it's got a dependency here. We built this pipeline so that this one depends on those other ones successfully finishing. And when they do, this one will run, compile that counts matrix with feature counts, and then do the statistical analysis. Okay, because this might not run at the same pace for everyone, depending on if your jobs entered the queue as quickly as mine did or not, I have sort of pre-baked output from one of these analyses that we can all look at together. So I'm going to go back to my dashboard. I'm going to go back to just plain old scratch.global. Since I'm already in a subdirectory of it, I'm just going to click here. Otherwise, I could click here. It's identical. Wait for it to populate the many thousands of directories. And then I'm going to go over here to filter. I'm going to type in capital TU. I see that there's a directory called tutorials. And if I didn't screw up anything, you should all be able to click on that. I guess I'll know instantly if yes, okay, good. I set the permissions okay. So now if we click on that RNA Seq tutorial, you'll see, similar to what we set up in our own scratch.global account, 2 different folders, one out and one called work. Most of what we care about is going to be in out. So if we click on there, we see a number of directories. And I'll point out that in the tutorial, this is all explained as well, both from the command line and when looking at it on Open On Demand. The ones, I'm going to focus this explanation on the ones that folks care the most about. First of all, when the pipeline is fully complete, we will end up getting an HTML file in that directory that is called something like bulk RNA Seq report. This is going to be the main output that we're going to look at. We're going to look at it in detail in about 5 minutes. And to look at it, you can't actually just click on it because web browsers are stupid and they assume that if you click on an HTML file, you actually want to see the raw HTML and not the rendered HTML. And so instead, you need to download it first and then open up the downloaded version. And that's going to contain almost everything that we need. But maybe you just want access to some of the intermediate files directly. Maybe you want to access the BAM files, which are the files that say where in your genome each of the reads mapped to. And those would be in that coordinate sorted BAMs directory. So we see actually links to a BAM file for each sample, and then a .bai file, which is an index file. And so if you wanted to look at where your reads piled up in your genome, for example, you could go back to that interactive apps list that I was showing at the very beginning, go to that IGV browser for Integrated Genome Viewer browser, and then navigate to your coordinate sorted BAMs directory and load up one of these and see in your genome where your reads piled up. These would also be the starting point for alternative analyses, such as differential splicing, or if you're going to do some variant calling from your RNA-seq data, something outside of the scope of bulk RNA-seq differential expression analysis. In the tutorial materials, it shows how to look at the head of one of those BAM files as well, if you want to poke around with that. The next directory is the counts directory. And this has a number of files that each contain, with one exception, the raw counts of the number of read fragments per each sample summarized to the gene level. All of these files that start with the word subread contain those raw counts. And if you're going to do any sort of statistics, those are the files that you're going to want to start with. I'll click on, or excuse me, not the summary. Click on this one. They look something like this. If you click on one, this time I clicked on subread counts.trimmed. It contains the following information. First, there's a comment line. And then there's 6 columns referring to where in the genome those counts belong to in terms of the name of the gene ID, what chromosome they're on, the start position of that gene, the end position of that gene, the stranded information, and the length of information of that gene. And then finally, the critical information for each of your samples, how many fragments mapped to that gene. So we see that some genes had 0, and then some had a positive number. And since these are counts, these are integers. In this case, since we're working with ensemble, these are in ensemble gene naming scheme. The difference between the trimmed subread and the subread counts is that besides the fact that it's about 6 times bigger for the subread counts file, is that the subread counts has extra information on each gene that says which exons in kind of a weird hierarchical tree format that frankly almost no one ever uses. So that's why we provided this subread trimmed file, which will usually be your input if you're wanting to do your own differential expression analysis. If you don't want to work with the ensemble gene names, but you'd rather work with the gene symbols, we also provide that here in the one suffixed gene symbol. But be aware that gene symbols are not always unique. So if you wanted to make sure that you had each gene uniquely identified, you'll want to start with the gene trimmed one. Finally, this one called CPM list here, this has non-integer information. So as part of the statistical analysis, I mentioned that the different samples might have different number of total counts per sample. And that, if we ignored that, that would really influence our interpretation. At the extreme, imagine that one sample had 10 reads total, and the other sample had 1,000,000 reads total. If we naively compared the number of reads in one gene between those two samples, we'd assume that it was much higher expressed in the second sample. So there needs to be a normalization process to deal with that fact and some other small finicky details which are sort of out of scope. And so that normalization gets applied to create what is called counts per million. When you do the statistics, the statistical software are all smart enough to incorporate that critical information under the hood, and they prefer to start with the integer counts because those can be modeled more correctly starting with that information. But for visualizations purposes, we usually want to work with the actually normalized data. So if you're wanting to just get your normalized data for visualization's sake, that's when you would start with the CPM list file.
Jeremy,
Yes.
can you look at the subgroup ones that you were looking at?
Yeah.
Just want to look at something really quick.
Which one?
Sorry.
Which?
Just a subgroup 10 counts. I think you look at that, yes, in there. I think the issue is that you have 6 samples in there, and there's supposed to be 8 samples.
Yep, because it continues right here.
Oh, it continues.
Yeah.
Never mind.
I'm
Never.
not sure why that word counts.
Never mind. Sorry.
Yeah, no,
Sorry.
Good.
I was trying to explain that word count that
It
we
was
did
a lot.
earlier. If it was
Yeah.
like missing 2 of them, like, oh, okay, that
Yeah.
explains. Never mind. Sorry.
No worries. The next directory here, DEGs, stands for differentially expressed genes. And this is only going to have something in it if A, you filled out that group template Excel spreadsheet, and B, didn't just fill out the first page, which had group information for each sample, but also provided a contrast in the second sheet that compared one group to another. If you did that, then there should be a file in here for each of your contrasts, as we see here. And this is the differential expression table for that contrast. And you could use this, for example, as input to making volcano plots or for pathway enrichment, as we'll be talking about next week. And so these files are arranged like this. They always start with the genes with the greatest evidence for differential expression at the top. In other words, the ones with the lowest false discovery rate corrected p-values. They tell you the name of the gene. This first numeric column is the log fold change. So that's how many twofold differences there are between your samples. This third column, log CPM, is the average expression in log 2 or in log 10. And then the statistical information, the F statistic, the raw p-value, and the corrected one. All right. Well, the rest of these we're going to not bother looking at because they are also in the report. So my guess is, I don't actually have my email open here, but if yours has completed, you'll probably have received an email saying that both the single sample jobs have completed with exit code 0. Exit code 0 means success. And also the run summary stats job has completed, hopefully also with exit code 0. But now, as I mentioned, the most important output is the actual report here. So I'm going to download it, clicking on the dropdown, hit download. I'm going to go to my downloads. I've obviously downloaded this multiple times. And now I can double click it from my downloads, and now it'll appropriately open up in the web browser. And you should see something like this if you do this. It'll tell you the date that it was run, some information about the pipeline. And now you can start to look at the critical pieces of results. This is all navigable with this menu over here. And it begins by saying what the data are that were processed. And so it's very good to check this to make sure that the input directory, the HiSat2 index, and the GTF were what you expected. If you went in and provided a group for each of these, it will give that group name right here. Otherwise, it will say null. And then really usefully, this bulk RNA-seq HTML file has the expression matrices embedded into them. So you don't even need to go navigate to that directory if you don't want to. You can just click on this and download those matrices yourself and start from there. Also, if you want to know exactly what was run, that pipeline file is also embedded into it. You can download it here. And similarly, the sample sheet. All right, but now let's start to look at these metrics and see if we can make sense of them. The first set of results are quality metrics, a handful of things to look at here. This first one, and there's one pair of columns per each sample, is the number of fragment counts. Now this is very low because we made our tutorial set small so that it would run quickly. But this number on the y-axis, the raw count, should correspond to what you were told from whatever company or core did your sequencing. It's how many fragments the sequencing of sequencing were done on your sample. The second column per sample is how many fragments remained after trimming occurred. So if you had a high quality sample like we have here, those two bars should be very similar in size. If the second bar is dramatically smaller in size, that could suggest that you had low quality read information coming off. Possibly it could suggest that the trimming settings should be adjusted. And you can ask us if you ever see that happen, and we'd be happy to help you interpret that. The next plot is the insert size metrics. And this information is gained by looking at the BAM file. And so in paired end data, they know once mapping has been completed, we know where read 1 went, and we know where read 2 went. And since we know where both of those reads were, we know how much space is between the beginning of the first read and the end of the second read. And that should map to your expectations based upon your kit. And in our case, the mean hovers around 300 bases, which is what we expect. For most of these plots, the information is available as a plot, or there's also a tab that you can click on to see it as a table if you prefer. In most cases where people are doing eukaryote, especially mammalian bulk RNA-seq, likely you are using a kit that starts by binding to the poly A tails of your fully matured RNAs. And so there shouldn't be a lot of ribosomal RNA present in them. If you're using a kit which does not allow for that, for example, a total RNA kit, then usually there will be a step where you're doing some sort of rRNA depletion. And this is important because rRNAs are the most common RNAs inside of cells, and usually we care the least about them, so we want to get rid of them. And so if you look at this plot and you see very high numbers, that could suggest that the amount of usable RNAs are low for your experiment. And you'll need to keep that in mind when interpreting the downstream results. And it could be an indicator that you need to change something in your wet lab processing for the next time around. I'll note that the cases where I tend to see high percentages here are when I'm working with a non-model organism that had to do some sort of external rRNA depletion, and often when it's coupled with also having low RIN values, which is a metric for RNA degradation. The next tab here gives the average quality scores. And really, as long as these are averaging over 20-ish, then you are probably pretty good. I like to see these in terms of the trimmed reads FASTQ style. If I look at this here, if I click on this graph right here, I see based on the position in the read on the x-axis, the quality on the y-axis, we see that it jumps up from 30, stays pretty high, and then starts to get a little bit lower towards the end. We see that the second read is a little bit lower than the first read, as that user observed earlier. But in general, these are all pretty high quality. We don't see weird zigzags or like real low qualities around 5 or anything like that. Okay. So this next chunk of graphs right here in this section QC based on mapping to reference genome can be very useful to look at. This first one tells us how many of our fragments, when feature counts looked at them, appeared to be in exons. And so again, this is going to be related to the kit that you used. If you used a poly A-based kit, ideally, the majority of your reads are going to be in exons. If you use something where introns were not necessarily spliced out before sequencing, then that number might be lower because you might have some intronic reads. If this number is very low, it might suggest that there was DNA contamination or something else went wrong. But here we've got it about 0.75, 0.8. That's just fine. This next one tells us the estimated number of unique fragments. So it's sort of related to the duplication rate. And we're seeing that the number of unique fragments is roughly similar to the number of actual fragments that we saw before. It was about 2 million per sample, which suggests that most of our reads are more or less unique, which is good. For bulk RNA-seq, we don't actually expect perfect uniqueness, because if you're sequencing a very common mRNA, that might actually be represented non-uniquely more than once. We see lowish duplication levels. Decent, it's fine. Then here is the one that I think is very useful to look at. gDNA and strand agreement. There was that funky metric where we had to type in for this kit's information -- strand space U to tell it that it's unstranded. If we did give it an RF, which is what it gives by default, or we changed it to FR, then what happens is it counts your data two ways: by being aware of the strands that you told it on the left, and by not being aware of the strands you told it on the right. If you picked the right setting, the left and the right, the distribution between the exons and the introns and the intergenic regions should be roughly the same in the strand aware and the unstranded. If you did not pick correctly, these graphs will look very different. Thank you. And that means you have to rerun it with a different stranded setting, and we can help you with that. This table is also very useful, or graph, excuse me, because it gives the distribution of your fragments that were counted in exons, and in introns, and in intergenic regions. And for RNA-seq, you're going to hope that you see something like this, where the largest bar by far per sample is in the exons. The next set of quality results are about alignment. So out of your read fragments, how many aligned mapped to your genome? And we're seeing that the numbers mapped here, and these are also shown below in graphs, is pretty similar to the number inputted, about 2 million, so that's good. We already saw duplicate information. This is just a repeat of that data. But this third column can be very useful. This is the number of fragments that had a mapping quality of 0. which means they couldn't be mapped to your genome. If that number is high relative to your input number, that may suggest you had a contaminant, and that there's actually more than one species in your data, and you should keep that in mind and consider it. These next plots tell us for each of these reads, how did they map? Did they map uniquely to one location best, which is what this darkest bar is down here? Did they map with equal quality to more than one location, which is what this small light blue, I think, bar is here? Did they map discordantly, meaning one read landed on one chromosome and the other read landed on a different chromosome? And then finally, did they not map at all? Did they have that map quality of 0? And so what you're going to want to see is that the uniquely mapped bar or chunk of the bar is the greatest proportion by far. That will indicate a good quality mapping. And these plots are shown both as plots in the tables, but then also if you prefer to just see it as a proportion-based plot right there. Okay. This next plot it's related to the previous one two ago about exons versus introns versus intergenic regions, but it says out of your mapped reads, how many of them could be assigned to a gene successfully? And we see in our case that the majority of the fragments were able to be assigned to genes successfully and then therefore counted into the counts matrix. So this is what we see. If we see the opposite pattern here, that would indicate something is up. This next graph tells us how many unique features, basically genes, are being detected with at least one read count in each of your samples. In human and in mouse, there's 30,000-ish unique genes, and usually only about a third of them are being transcribed at a measurable rate with typical depths of bulk RNA Seq. And that's roughly what we see here. We're seeing about 10,000 genes are being counted with at least one fragment per sample. So that's good. If this was very, very low, that could suggest that you're only capturing very highly expressed genes for some reason, or it might be related to a problem in one of those other graphs, like maybe you had contamination, or maybe you're getting too much intergenic mapping. Okay. The next set of plots are actual statistical information on your samples. The first one is just the number of counts per million in all of the genes shown as a violin plot per sample. And this is a very typical pattern where most genes had very few counts for them because they were lowly expressed. And then there's a long positive tail, meaning that there are some genes which had much higher expression than all the rest of the genes. But probably what you care the most about are these next two graphs, the sample clustering heat map and the multidimensional scaling. The sample clustering heat map, is shown here, and this is also available in that plots directory that we briefly looked over earlier. And this took your counts data, normalized it, found the top number of variable genes, grabbed those genes which showed variation in counts across your samples, and then hierarchically clustered your samples by how similar their expression was to each other. And if you provided group information in that group's template, then that group information is shown as a color up here. So this particular plot was not sorted. We did not tell it, put the bone on the left and put the spleen on the right. Based upon the expression information, it it clustered the bone separately from the spleen. That's useful information. That tells us that it seems like expression is differing at the highest, the greatest difference in expression is occurring between those bone samples and between those spleen samples, because that's where the tree branches first. And then we see much smaller branch lengths between samples, so replicates seem to be relatively similar to each other. A graph like this will suggest that you will indeed get some differential expression. If all of your groups are interspersed with each other, you're less likely to have a strong biological effect in your data and therefore less likely to see differential expression. This third graph says multidimensional scaling, but in truth, this graph is actually just principal components analysis. Multidimensional scaling is a generalization of that. We find principal components analysis the most directly interpretable of those, so we usually stick with that. What principal components analysis shows is for this graph, which shows the first two axes, it summarizes the data by finding the genes which describe in a linearly combinable way the first the largest amount of variance explained, and puts that on the first axis here. And group of genes linearly combined, which explain the second highest number of variants, it smushes onto the second axis here. And if that didn't mean anything to you, don't worry about it. What you want to look at on a graph like this is you want to see, similarly to that clustering heat map, do your groups go in separate locations, suggesting that there are large differences between them, especially on this first axis here, which is the largest separation in data. And we see that here. Our spleens are on one side, our bone marrow are on the other. We also see in this particular graph that there is definitely variance between the samples showing up in the second greatest axis of variance. It's not too big of a concern, but something to keep in mind that maybe your replicates here are rather widely distributed. This particular plot can also be really useful for looking at batch effects. If you read the instructions for chirp in more detail, in addition to group, you can actually add a third column for batch effects. And if you see that your batches are separating in this plot, that could suggest that you need to deal with the statistics a little bit more carefully than is done with just the automated chirp submission, and then you should talk to us with the help desk ticket. All right. Finally, if you provided that differential expression contrast sheet in the Excel spreadsheet, it will show you any differentially expressed genes here in different tabs per each contrast in this location. You can, of course, access the full list in those files that I showed you before. Then finally, below it, it tells you methods that were applied to each of your steps and also where all of these outputs are so that you can access them again later. All right. It's now noon, so I'm just going to very briefly explain some other things that you could do. But I'd also like to point out that somewhere in here, I don't want to spend a ton of time searching, somewhere in here it shows how to load the counts matrix that we provided into R through the supercomputer if you wanted to start to do your own analysis yourself. But so some other things which could be done downstream of this are you could start with the statistics files, make volcano plots, or do pathway enrichment, which I'll tell you how we do next week. You could start with the counts themselves and do things like weighted gene correlation network analysis to see which genes have similar patterns of expression across all of your samples. We could start with the BAM file itself, visualize them in IGV, or do other things like differential splicing or even variant calling if your data are good enough for that. All right, well, that's pretty much right on time. Hopefully this was useful to you. You should now have your outputs from this test in a directory on MSI. If you're completing the badge process, then I know that submitting that information will be part of the badging process. I'd be happy to stick around for a few minutes and answer any questions that anyone may have. I'll just remind you again that we have our pathway analysis tutorial next week and the single cell tutorial the week after that. Thank you all very much for listening to me speak at you for 2 hours. I know it's a lot to cover. I appreciate your patience and also answering questions. And thank you very much.